The first stage of transcription that we are going to talk about today is initiation. In bacteria, the process starts with a promoter region containing several important sequences called consensus sequences that allow for the transcription apparatus to bind. A few common consensus sequences are the -10 consensus sequence, or the PRIB-NOW box, the -35 consensus sequence, and the general upstream element. The placement of these sequences is important because they set the starting point for transcription. Proteins can interact with or around these consensus sequences to either enhance or inhibit the transcription process. The sigma factor associates with the RNA polymerase to form a halo enzyme that recognizes and binds to the promoter. Different sigma factors can recognize different promoter regions. The halo enzyme then unwinds the DNA via its helicase activity abilities, and a complementary RNTP forms the first hydrogen bond to the first base at the transcription start site. As the RNA polymerase leaves the promoter region, the sigma factor releases and transcription enters the stage 2, elongation. However, the process of initiation in eukaryotes has several variations. Before any enzymes may bind to the DNA, the euchromatin must be modeled in such a way that the DNA is accessible, usually through acetylation by an acetyltransferase to form euchromatin. Once exposed, transcription factors assist in recognizing the promoter sequence similar to the sigma factors. The basal transcription apparatus, which is the RNA polymerase and the transcription factors, then binds to the core promoter immediately upstream of the gene. The core promoter may have one or more consensus sequences, often including a TATA box. Immediately upstream of the core promoter is the regulatory promoter, which bends to interact with enhancers through the transcription activator proteins. After the RNA polymerase has transcribed about 30 bases, elongation begins. Many of the transcription factors are then released and move to initiate transcription of another gene.